Welcome to Environmental Law Explored, a podcast series, the podcast of the American Bar Association section of Environment, Energy, and Resources. SEER is a member organization whose mission is to foster the success of a diverse community of environmental, energy, and resources lawyers, advisors, law students, and decision makers, and provide a premier forum for the exchange of ideas and information.
This is episode number 4 in the Microplastics series, which is covering the ecological risk and current ecotoxicology. If you missed it, the prior episode covered sampling methods and analytical techniques. I want to welcome our presenters and allow them an opportunity to introduce themselves. Dr. Lance Yonkos is at the University of Maryland, and Shannon Edmonds is a project manager for Roux. Lance, please give us a little bit of your background.
Okay, well, certainly I am an associate professor at the University of Maryland. My background basically involves looking at the sources and fate and transport of contaminants into the environment. Of recent, it largely involves urban environments and rivers. And so microplastics fall comfortably in this because they are human sourced, and a lot of our activities introduce them into our receiving waters where they encounter organisms and so forth.
Thank you. And Shannon, if you will give us a little bit of information about yourself too, please.
I'm Shannon Edmonds. I'm an environmental scientist at Roux, where I mostly work in our litigation and ecosystem services practice areas. So microplastics kind of ties both of those together nicely. I've been involved in our microplastics special interest group for the last year and a half or so, and Lance was my advisor in grad school, so that's how him and I know each other.
Fantastic. Well, thank you both for joining us today. Lance, we'll start with you. Can you give us a little bit of information about, when I say microplastics, what does that mean to you? What do you think of?
Well, I've been doing research on microplastics, so I have a pretty good understanding of them, although I'm constantly surprised as I read more. Basically, we manufacture lots of plastics used in lots of applications, and when they are at their end of life, depending on how they were handled, they tend to make it into the environment. And if they are progressively degraded in some fashion to a very small size, less than 5 millimeters is the sort of the current criteria, then we describe them as microplastics. From there, their behavior, their potential interactions with organisms, and the consequences become kind of my area of interest.
Thank you very much, Lance. Shannon, would you like to kick us off with the next question or topic?
Yeah, so listening to the news, it seems like microplastics are pretty much everywhere in the environment. Lance, what do you think about that?
Well, it's pretty much true. We could probably search and find some places that we didn't find microplastics, but the trend has essentially been, when we first became aware of them, largely as we looked at ocean gyres and beaches where large macroplastics and then progressively smaller and degraded plastics were found. And we began to start looking progressively into urban rivers and into lakes. Wherever you look, you find them. Obviously, we're now starting to collect from deep ocean samples, not just in aquatic systems. They are commonly found now in urban soils. They're found in agricultural soils. Just a few years ago, they were reported to be found on the Alps, where they were obviously airborne and blown there, and even most recently deposited in fresh snow in Antarctica, which makes it clear that they can be atmospherically transported very long distances. And so there's no place on the planet that is likely to be immune to their arrival and potential impact.
So unlike other contaminants, like maybe metals, where we might expect a background level or a natural presence in the environment, these are really going to be the results of human activity.
Sure. I mean, obviously they are human manufactured and used, and there is no natural source for these materials. So with that in mind, there's a very strong correlation between population density and urbanization, percent imperviousness, and so forth, and the abundance of microplastics that are found in nearby estuaries, for example.
I seem to recall a 2014 paper that you actually wrote about that in the Chesapeake Bay.
Oh, you're too kind to mention that. Yeah, so I did a small study. I don't want to take too much credit, but investigating the abundance of microplastics in relation to Baltimore Harbor, for example, as an urban center and industrial center versus more suburban and agricultural and remote regions. And we supported what a lot of others have found, that not surprisingly, where the sources are, that's where you're going to find the greatest abundances of microplastics.
So this is mostly a problem about poor waste management?
That's where I began. So I'll say yes and no. Absolutely. The way in urban environments, the way we have waste treatment and sewer systems that are combined, for example, we introduce a lot of plastics and microplastics in the influent, and these systems aren't designed to deal with that. So even though they're in many cases fairly effective, they become a point source of discharge. And obviously our trash management can induce a lot of macroplastics to the environment if it's poorly done, which are then fragmented and degraded. But it's probably also important to keep in mind that a lot of sources actually are unintended consequences of sort of normal intentional practices that we perform in the use of these materials. I understand tire wear has been mentioned previously. This is an enormous contributor to the abundance of microplastics. It is, by design, every tire ever used in a vehicle lasts until the tread's worn off. And that means it's introduced to an impervious surface that has washed it off into a nearby waterway, most likely. But other things we could think about, aquaculture. We think of these as solutions. We need to produce food. Fishing the oceans to depletion is a problem. But if you think that all of our food is now produced in open ocean pens, that every aspect of that is made of fishing lines and nets and cages and floats that are plastic, and their functional life is until they're no longer able to contain the organisms or float. And that means they've calved off literally millions and millions of tiny plastics directly into an aquatic system. Even green infrastructure, we're really hot now to control urban water runoff and so forth. And our solutions, and they should be applauded because they're very good, are grass filter strips and receiving and retention ponds. But then our very first thing, after they've grown a lot of grass in them, after they've accumulated a lot of trash and plastics, is to run through with a lawnmower and turn a plastic cup into 10,000 pieces of plastic that then wash pretty easily away. So these are manageable, but it is the way we tend to get things done. And a lot of other things fall into this. The number of blue tarps that were sold after Hurricane Katrina was something like 15 million. Everyone in the United States was shipped down there, and they were put on the roofs of every house, and then they shredded, and blue fibers became the thing that was detected most abundantly after that event. And so artificial turf and road markings and sort of anything that's normal life cycle is to be placed in the environment and to physically degrade becomes a source of potential concern.
So since these are basically everywhere in the environment, they must pose a considerable threat to organisms living in all these different compartments.
Yes, they do. So they are available to encounter organisms and vice versa. These risks have to sort of be categorized. The most obvious involves physical interactions with the plastics. And this is really where this whole field got started. The NOAA Marine Debris Division was tasked with looking at marine entanglement and encounters with marine debris, which included plastics. And so, of course, early on, to the scale of this, these were not micro, but macroplastics, the idea of whales and turtles being captured in ghost nets. But the scale works with this all the way down. So as you progressively look to smaller organisms, even tiny zooplankton and larval fish, these become entwined in microfibers. It inhibits their mobility and their opportunity to feed properly. What I consider a startling discovery, this is sort of my new hot topic, is the report of just the sheer abundance of microfibers that are currently entangling coral polyps in sort of any area that's in proximity to a population. And if we imagine that these corals and the associated reef communities, these are the ones that we're kind of most worried about right now for all of the other reasons of climate change and sea level rise and ocean acidification. So when you add stressors like this that potentially limit their feeding ability, obviously this could lead to pretty profound effects. As we get progressively smaller, the concern tends to switch from entanglement to ingestion. And this is we've seen a lot of headlines with birds that have ingested comparatively large plastics, and the pictures are always very alarming because they're colorful, and it's a bird who has been opened up, and we're showing these things. Again, this works at pretty much any scale as well. And so as you get to progressively smaller materials, you're going to have more organisms that are able to actually ingest them. Within the opportunity to see these things with marine mammals, for example, in virtually every instance of a marine stranding of a marine mammal, they have found substantial loads of plastics, and including microplastics, within their digestive tracts. Seabirds, with very few exceptions, when they are found, they harbor these in their guts. Commercial bivalves. So when you think about filter feeders, obviously they're going to be uniquely vulnerable to capturing small floating particles and other materials. Another slightly, maybe not surprising, but new in the news, the notion that organisms down in deep ocean trenches, for example, there's an amphipod species found at about 35,000 feet. So this is 6 miles deep within a deep ocean trench. And the majority of them actually harbor microfibers within their tissues. And so they're obviously able to transport these distances and become incorporated. And then obviously the concern here is that consumption of these is going to clog their digestive structures in some way, and lead to false satiation or some sort of reduced feeding capability.
So basically eating microplastics instead of food, but then feeling full.
Exactly. And so if you're inhibited in your ability to consume food, or if you are eating things that are non-nutritive, that's going to reduce your total feeding success. And this may not be that big of an issue for a lot of organisms. Oysters, for example, are very good at capturing all sorts of things as filter feeders, and they're also very good at sorting them and spitting them out. So maybe this isn't a big problem, but if you think about particularly sensitive life stages, larval oysters, for example, have a very short window to feed themselves copiously on algae and grow, and then find a hard substrate to set on. And if they don't get there in a fairly tight window, they won't make it. So they're going to be sensitive. If you think about larval fish, they need to grow very quickly. If they have a handful of plastic beads or some other things in their gut and it inhibits their growth, that's going to dramatically increase their likelihood of predation and not thriving to the point of adulthood and reproduction and so forth. And this is going to go for any number of species. There's some other concerns. If you think about ingestion of these structures, they're possible sources of pathogens. Microplastics, the nature has whatever constituents are normally there, but if you add something new as a possible substrate, it can be colonized with organisms, and that's possibly a good thing. They form these microbial biofilms of bacteria and fungi, but because they are durable and because they are often buoyant, they can be transported significant distances. So discharges that might be coming out of wastewater treatment plants or simply urban runoff with associated animal waste and so forth, these can actually incorporate the pathogenic varieties of bacteria and so forth into these biofilms, which are then able to be transported significant distances to where organisms might encounter them.
And I guess the biofilms might even make the microplastics tastier or maybe even more attractive to organisms to then ingest to kind of continue the problem.
Well, that's exactly true. I mean, most things that are filter feeding are looking for something to eat, and probably nothing tastes better than some of the chemical cues that are coming from these materials with a coating of a variety of bacteria and so forth on them. So it can be an attractant, absolutely.
And have you seen evidence of transport of invasive species too with microplastics or kind of similar?
So there is evidence of this, and it goes right along with the rest of this discussion that in macroplastics and large things that can transport long distances, the same applies. The scale of these, the real challenge is if you go out looking for something tiny in the environment, you have very little likelihood of knowing where it came from. But there is no doubt that these have a very long environmental persistence. They can be windborne, they can be if they float, they can definitely be on surface films that are transported long distances. So without a doubt, anything that's that durable in the environment has that opportunity. They can also, this is a bit of a stray from the topic, but they can be taken from harbors in one place in the process of ballast water collection, for example, and it deposited in others. And it's sort of a hot topic to try and treat invasive species in that. I don't know if the microplastics constituent complicates that in any way or not, but it's certainly a possibility.
So I would think one of the greatest concerns is probably the possibility of toxic effects associated with microplastics exposure. This also seems to be one of the areas of greatest uncertainty. Can you elaborate on that?
Well, that's exactly right. Like everything in this, it is a complicated discussion when we talk about microplastics. One of the challenges when we're really trying to address the toxicology is that conventional environmental toxicology isn't all that well suited for looking at microplastics. And the reason is it evolves kind of from human toxicology originally, and the idea that contaminants should be thought of as molecules or even maybe individual elements. And so then we say, well, what is their behavior in the environment? Are they likely to want to float around in the air or dissolve in water, or are they going to stick to soil or other things? And this is going to determine their likelihood of exposure. And then from that, if you can become exposed, are they available to come into contact with membranes, either in the digestive system or the respiratory system, and then be taken up into cells? And so this field of study has spent 5 decades building that model. And all of a sudden we say, well, we want you to look at particles. And these are a physical thing. These are made up of lots and lots of polymerized molecules. And so it doesn't fit comfortably into that paradigm because it involves a bit of a deep dive into material sciences, and it really involves an appreciation of the mechanisms of transport and weathering and breakdown. So it's not to say that these are foreign to toxicology, but it's to say that it's an incremental process to learn how they participate in this whole conversation. And then, of course, the second one, and maybe the biggest, is that we say microplastics like it's this one thing, and it's actually this enormous constellation of different materials, and even with the same materials, different times when you encounter them, the sizes and the conformations and configurations. And so all of these make it really difficult to say, all right, let's just do some toxicology and see if we can pick up a result. And so with that in mind, there's some good review papers out there, and they describe sort of the current state. A good number, maybe not a majority, but a good number report toxicological effects of microplastics exposures. And I'll talk later, maybe more specifically, but they mention categories of leading to shortened lifespans or reduced feeding success and slower swimming, maybe changes in number of offspring being produced and disease frequency. But in truth, there's actually more studies out there that don't detect any kind of deleterious effect. And even some of them indicate improved performance in a lot of these measures associated with microplastics exposure.
So what do you mean by improvements? Like increased weight or more young, or why is that happening?
Well, so exactly. The idea is you set up a study, you have control groups and you have exposed groups, and you look at a result, and sometimes the exposed groups do better. And the consensus generally is that in a laboratory, you try and limit certain things, and sometimes that includes feeding opportunities. And so if you all of a sudden make this substrate and it grows these biofilms, you may actually be providing them increased nutrition. And whether that would be true in nature or not is not exactly knowable. But the notion is that it isn't a simple thing to just set up a study and come up with a result and defend it. And so there are many of them that are indicating that.
So looping back to one thing you said, that lots of these microplastics are toxic, and there are reports that have come out on that. Have you seen companies and manufacturers kind of taking notice of that and thinking about or even actually changing the types of plastic products that they're producing, or not yet?
I wish I was more versed in that. What I will say is there is a pretty big push right now towards greener approaches in plastics that are being used. And the idea is more, in my mind, about coming up with polymers that are more amenable to molecular recycling. So right now our approach is we throw it all out and they sort it, and there's some things that can be done, and if it's contaminated, it all has to get incinerated. But there are newer approaches that are much better at taking a complex mixture of different plastics and largely disassembling them at the molecular level. So if there's some that aren't able to fall into that criteria, they're probably, people are saying, well, let's see if we can find ones that will. And that's great, but that still doesn't necessarily, that just incentivizes recycling. It doesn't really address the idea that if we can't control our trash and it makes it into the environment, that it's still going to degrade and become microplastic. I think we're a long way from saying that a corporate decision to change from one thing to another is going to have a meaningful change in the microplastics that end up out in the environment.
Gotcha, thanks. So at least we know that these risks are out there, and then maybe the next step is for people to take notice of them and make some substantive changes.
Absolutely.
So oftentimes I feel like people mention the possibility for microplastics to be carriers of other contaminants or pathogens. Do we have enough evidence to know if that's happening?
Well, we do. So first, if we start with the idea that the microplastics are there's lots of different possible polymer makeup of those, but then they tend to be a complex mixture of additives. There's a lot of reasons for trying to modify them to perform their purpose. So plasticizers to change their rigidity and durability, and colorants. In a lot of cases, we intentionally put things like flame retardants for obvious reasons, maybe not justifiable, antifungal agents to reduce degradation when they're actually in use. That's common in caulks and other materials, but these are designed to be toxic. And so these can be associated with the plastics and the microplastics. But then once they're in the environment, as I said, they are a durable substrate. And therefore, because they are organic in nature, compounds that are uncomfortable in water but are environmentally durable will have a habit to become associated and absorb and accumulate on and in them. And this becomes that classic suite of everything that we really worry about in environmental toxicology: PCBs and dioxins and brominated flame retardants and organochlorine pesticides. These are exactly the kind of compounds that, because they're uncomfortable in water, they're looking for an organic matrix to attach to. We, in fact, in trying to study the abundance of these in nature, we take little bits of plastic and we deploy them as passive samplers, and then we retrieve them 2 months later and we measure what's been on them. So it's clearly demonstrated that they accumulate substantial and, in fact, representative amounts of the contaminants from the environment they come from. And of course, what that means is if they're floating through a wastewater treatment plant, or if they are sourced from an industrial or intensely urban environment where they might be associated with contaminated sediments and soils, they can accumulate these contaminants. And then obviously they're durable and they're buoyant and they float, and so they can then be transported to places where they might be ingested by organisms. And then, of course, if you take all of this together, the challenge is the toxicological profiles of the polymers themselves, the additives, or any of these other things, how they leach, what their environmental durability is, and weathering. All of those are going to differ. And so it makes it complicated to make a statement about what the toxicity of a particular particle or constellation of particles might be. And sort of related to this, we mentioned from the beginning that everybody started looking at large stuff and progressively smaller, and so size is really critical with this. Smaller particles are much more easily consumed by smaller organisms, and as we continue down, the smallest particles that we still define as microplastics, maybe down in the several micron in size, these are actually able to be translocated across cell membranes. So that classic toxicology about something has to get close to a membrane and then maybe get inside a cell, this is where we get to that, where they actually move into tissues, and they can be, in cases, transported around within the vascular system. So even though the exposure might be within the intestine or across a gill, or for terrestrial animals, it could be within the lungs, they can then be moved to the liver or into muscle and into the brain, for example.
And I'd imagine the smaller particles that are the most mobile and most ubiquitous and are the ones that are translocating are probably also the hardest to remediate.
Exactly. And more than that, they're also the hardest to do any research on.
Yeah.
It's hard to find them. If you go out in nature and you drag a net a 2-micron net, you catch an awful lot of stuff that you have to sort through to find the plastics. And so there's a lot of challenges associated with identifying them and enumerating them. And certainly at that scale, determining polymer types and things like that are very complicated. And even with that, shape matters. Fragments are and especially fibers are being found. Fibers are being found to be some of the most abundant of the materials out there. They're also being found to be some of the most problematic for organisms. And first up, it probably relates just to that surface area. The irregularity and the elongated materials tends to allow them to transport more easily, to be more mobile, but also more surface area. So the leaching, the attachment and absorption of contaminants to them, and then the leaching of contaminants off of them once they're maybe within a digestive structure can be higher. And then these are just problematic shapes. They're more likely to become lodged within the digestive tract or within respiratory structures. I think one of the models that sort of informs us a little bit here is if you look at asbestos. There's a tremendous amount of science on this. There's nothing about the molecule or the makeup of asbestos at a molecular level. It's the fibrous morphology that is actually small enough and sharp enough that's able to cause nuclear and DNA damage and lead to tumor generation. So I won't say microplastics are causing tumors, but I will say that the shape of particles is going to have a lot to do with where and what tissues they can get into and how long they're going to reside there.
And those fibers are also the ones that are most likely to travel the furthest by wind, so that could be concerning.
I believe that is correct, yeah. And the issue with laundry and the idea that our polar fleeces are just calving off measurable amounts that are going to wastewater treatment plants. And again, they're reasonably effective at capturing them, although most of that then ends up going into biosolids, which are preferentially now land applied. They are a great soil amendment. They are good for agricultural purposes, but they come with some of these other potential consequences.
So what are some of the best understood examples of microplastics associated toxicity?
First up is the most obvious, that if these things are going to get into the digestive system, they have the possibility of just causing frank damage to the intestine. And the cascade of events associated with that is reduced feeding efficiency, limited acquisition of nutritious prey, which affects metabolism and energy allocation and behavior, ability to avoid predators. Also increased opportunity for pathogenic infections. Even if those infections aren't sourced from the plastic themselves, having lesions and abrasions and so forth, internalized or external with entanglement, can be a problem. There's a lot of studies with shellfish, with zooplankton, things that tend to be filter feeders, that have shown that reproduction is reduced. And for the most part, it appears to be about this energy allocation. They're simply not getting enough food in to source it for producing viable gametes, producing useful eggs and so forth. But there's also some studies that are starting to demonstrate that the constituents in the plastics themselves, and it's often not the polymers, but some of these plasticizers and phthalates and some of the other materials that are added are estrogenic or at least hormonally active in some way. And so there could be elements of endocrine disruption that are part of this. There's clear evidence of once microplastics actually get inside tissues, that they lead to sort of a generalized inflammatory response, which leads to oxidative stress. It's basically an abundance of free radicals that lead to profound cell damage. It can lyse the cell membranes and it can damage the nucleus. And essentially it leads to poor cell function and premature aging. So these are hard things to translate into a toxicological effect of consequence in nature. But if organisms aren't going to thrive and live to their full life expectancy, at some level, that's probably going to have some impact at a population scale.
You've made me think of two different points here that I'll chime in with, and these may not be relevant or might not have background, but I saw an article recently about, I think it was mealworms that can eat certain types of plastics or styrofoams or things like that. Do you think that's a positive thing or a negative? Should we be looking for more ways to kind of dispose of plastics with something like mealworms? And the second part of that question, we've talked a lot about animals, but have you seen these microplastics in plants? So for, like when the waste is applied to like fields and things like that, is corn going to have microplastics when I'm eating that?
I am not a botanist, I'll start with that, but I will say that the good news with plants generally is that they are great at absorbing dissolved things through their root rhizomes. They're not so good at capturing particulates. And so, that's helpful because that means that the chunks themselves are unlikely to make it into and transport in plants, unless you've read something that says otherwise. But the flip side of that is that if they're sourced from biosolids and they're bringing a variety of materials that we don't want with them, those can become desorbed and taken up by the plants. So there is the possibility that contaminants, we measure things on average, at a wastewater discharge and in biosolids. And if we have this rarefied individual variety of components that are very much enriched in contaminants, it's possible that they become placed into the environment in a way that we're not really all that aware of. Agriculture has a lot of concerns that fall in that category. I don't necessarily put the microplastics making it into plants as a main concern, but I do say that the application of these into agricultural landscapes has the possibility of being introduced back into the environment through terrestrial sources. Worms are food for birds. So worms eat soil, they get plastics that have all these bacteria on them, birds eat worms, and there are some demonstrations of food chain transport like that in terrestrial systems. I just read an article where they were reporting how crickets are eating plastics in houses, and then they're eaten by frogs, and they're finding transport into frogs. So the more we look, the more we're going to find, and there will be some surprises in there, without a doubt.
And that also sounds like it could be a possible source of risk to humans when you think of trophic transfer and eating fish.
Sure, exactly. So there's kind of a lot of places where this applies. If we apply these materials and they get taken up in these zooplankters and so forth, that would not be a typical part of the human diet. Obviously, even if we don't see classic bioaccumulation where the chemicals within them are going up, big fish eat little fish and little fish eat these. And so eventually it gets to something that's in our food chain. And so there is an element of human risk from things that are sourced that way. And in truth, this is already reasonably well understood. We look in hatchery production. Again, everything done there is done with plastics for the reasons of their durability and so forth. And so there is an understanding that there is consumption of these, but even in harvested fish, we like big fish, we like old fish. That means they've been out there a little bit longer. They've had more opportunity. And especially as we look at these smallest plastics that are able to translocate into tissues, they're going to be increasingly high in the muscle, which is the part that we actually eat.
And I think one point here too, I mean, these microplastics, when they are absorbed into the tissues, they stay there permanently. They don't get flushed out of the system later on. Is that correct?
That's exactly right. There is no real mechanism that organisms are designed to deal with this. They can encase them in some macrophages to form a little cyst to control them if necessary. Actually, that's something I hadn't mentioned previously. There is evidence of interactions between microplastics once they've gotten into these cells and the immune system. And so they are recognized as a foreign body, and they can be attacked by phagocytes. They can induce an immune response, but also if they're so ubiquitous, eventually responses decide to be attenuated. And so they can actually reduce immune competence to responding to other pathogens or real pathogens. And as a consequence, there appears to be a relationship between prolonged exposure to microplastics and increased disease incidence from secondary exposures to pathogens.
So it sounds like there's a lot that we should be worried about. How worried do you think we should be?
Are you an optimist or a pessimist? Depends on what day we get you.
Exactly, yeah.
Well, I'll start off by saying that even though we've been looking at this for as many as maybe 15 years now in a serious way, we don't really know the true extent of this problem. The vast majority of the research has been on marine species. Certainly, there's a lot more attention now happening in freshwater species, but it's incremental. With the exception of marine birds, there is very little terrestrial research on species. that have been studied. And so that's one limitation. If you think that some species are robust and some are sensitive, we haven't looked at enough now to make a statement on that. And then the next up is that almost everything that we can realistically call a toxicological study has been conducted under laboratory conditions. And for very good reasons. We need to have control so that we can investigate what the likely effects are, but they don't then necessarily reflect real world exposures. It's been our practice to use these neat compounds where we have a single polymer, maybe it's in a nice spherical bead, and maybe it's actually been fluorescent labeled in such a way that we can see where it's going. And we do a series of sizes and concentrations of these, and we look at them in relation to a variety of organisms, and we report back the kind of results that I've said, that they reduce feeding and that they move into tissues. And while all of that's true and all of that's very likely to be happening in the environment doesn't have these nice, clean, unweathered materials. They are a complex mixture of lots of different plastics of different configurations. They've been weathered in ways that they, and especially if intense sunlight exposure is part of this, the photolysis can actually bioactivate and modify molecules that never would have been part of the compound to start with. And so the laboratory tests that have used these don't necessarily inform us of what we're going to see if we go out in nature. And that means that there's a lot more work that's left to be done. Add on to that the idea that when we do a lab test, I said half of them didn't find effects or more than half, but we were using really happy animals. And we fed them and we controlled their temperature and we kept predators away and we did everything to make sure that we were only looking at the stressors of our particular contaminant of interest. And in the field, it's not that simple. They are constantly pressed with the stresses of sufficient food acquisition and predator avoidance and dealing with parasite and pathogen defense and whatever the energetic demands are of adequately producing young for reproduction. And so it may take a trivial and not detectable insult of these exposures that actually leads to a catastrophic result. And so we're not in a place where we can say that yet because it's difficult for us to even find these tiny particles in the abundances and varieties and so forth in biota. And teasing it apart from all of the many other things makes it difficult.
So now we have touched on a lot of topics in this conversation. What is the one main thing you want the audience to take away from this today?
I think the take-home message is that it's a slow process of study, but it's necessary, that there's a lot more to do. Microplastics, there's nothing on the horizon that suggests this problem is going to diminish in the next few decades. And even if we do a very good job of reducing our introductions of new materials, this act of weathering and degrading and transporting is ongoing, and so will continue and will likely increase for many decades. So with that in mind, we maybe don't have some really scary smoking gun that says, everything's going to catastrophically collapse, but it's important for us to keep looking because we don't even know what we don't know yet. And I bring up the case that corals, as an example, are very much imperiled, and they are becoming an interesting area of study. But even more than that, the idea that we're going to go to a deep ocean trench somewhere that we've never been, and we're going to send a submersible down there, and we're going to take some video, and we're going to see plastics, and we're going to capture organisms that are unknown to science, and we're going to open them up, and they're going to have plastics in their intestine. And there's no way we're going to do toxicity studies of these. It's just not the way it works. So we don't even understand the nature of some of the risks that are out there. More time and more investigation is necessary. I think the consensus is going to be that many animals are robust to this, but a handful are particularly sensitive. And if it turns out that there's a variety of polymer types or additives or other constituents to these that are the real problem, then we can focus our attention there. And that can be where we try and affect the fastest and most consequential changes.
So lots more to come. We don't know what we don't know. Well, thank you very much to our presenters and to the ABA. The next episode will cover health risk and current human health toxicology. So be sure to tune in. Thank you all.
We hope you enjoyed today's episode. To learn more about our section, please visit our website at www.americanbar.org/enviro. Please check where you found us for future episodes. Thank you for listening.